Hello and welcome to Stars, Cells, and God, the show where we discuss new discoveries taking place at the frontiers of science that have theological and philosophical implications, as well as new discoveries that point to the reality of God's existence and God's character attributes and plans. My name is Hugh Ross. I'm joined by the president of Reasons to Believe, Fuz Rana. I'll be your guide as we explore a couple of interesting topics. Fuz is going to talk about a new organelle discovery and its implications for the history of Earth's life. And I'm going to be talking about the atmosphere and the atmospheric jump that enabled animals to show up and what we now know to be the cause of that. But before we get into that discussion, I want to encourage you to subscribe to our Reasons to Believe YouTube channel and click on the bell icon so that you can be informed of our new videos. Learn more at reasons.org or by following us on social media at RTB_official. Well, Fuz, we had a little bit of a talk about this intriguing discovery you've uncovered, and I think it's going to have some significant implications theologically, so jump into it.
Yeah, well, sure thing. And to get things started, I don't know if you know who this is a picture of. You might, actually. He actually has the same heritage as you. He's Scottish.
He's Scottish. Well, if you go back a century or two, a lot of scientists were Scottish.
Well, he's actually not a scientist.
He's not a scientist. Okay.
Well, actually,
I'm not familiar with this individual.
His name is George MacDonald.
George MacDonald.
So he was born in 1824, died in 1905. He was a Scottish minister. He was part of the Congregational Church. Probably had more liberal theological leanings, but he wrote a number of books on theology. Also, a book that he published was a collection of his sermons. But he's actually a poet, more known as a poet and an author. In fact, he's considered to be the father of fantasy, of the fantasy genre. So he played an instrumental role in encouraging Lewis Carroll to publish Alice in Wonderland and was an inspiration for C.S. Lewis and J.R.R. Tolkien in the work that they did. But in one of his novels called The Princess and the Goblin, he has this interesting statement that actually now is a fairly famous quote attributed to him, where it says, "Seeing is not believing, it's only seeing," right? And so, it's actually quite profound because it means that just because you see something doesn't mean you have a full understanding of what you actually are seeing, right? So he kind of turned that whole expression of seeing is believing on top of it.
That shows up in a whole bunch of science research papers at the introduction. So
yeah, well, there you go. I didn't realize that, but it doesn't surprise me.
Right.
It makes sense. Well, that theme of seeing is believing is actually one of the arguments that I hear evolutionary biologists often use to justify the evolutionary paradigm. They'll say, look, we can see evolution happening right before our eyes. Seeing is believing. And that idea was the theme of a book published maybe 20 years ago now by a guy named Stephen Palumbi called Evolution Explosion, where he argued that, look, we see all kinds of examples of evolution happening, again, before our very eyes. All of the examples were microevolution, speciation, or microbial evolution. And he even argues, look, we are actually causing much of that evolution to happen because of our lifestyle as humans. And so, and that evolution has some very real economic effects. And to ignore evolution is really doing a disservice to humanity. So it's a very interesting argument. But that same mindset seems to be permeating a recent discovery of a new organelle, as you mentioned, in an algae. And this is an example of a headline that this discovery triggered. This was a paper published in the journal Science in early April, but that inspired this Science in the News piece by Michael LePage, where he said, a bacterium has evolved into a new cellular structure inside algae. So it gives the impression that we literally are seeing this happening right before our very eyes.
And not just microevolution, they're actually claiming this is a major change.
Yes, yeah. So this is in effect considered to be support for an idea called the endosymbiont hypothesis, which is a central idea in evolutionary theory describing the origin of complex cells or eukaryotic cells. But for fun, this is an image taken from the New Scientist piece showing that organelle that's now being dubbed a nitroplast. But the idea was that this was actually a cyanobacteria that had what's called an endosymbiotic relationship with an algae.
Can I stop you to define your terms for people who may not
Yes.
be biochemists?
Yeah.
So,
Yeah. So, well, and we'll get to that in a second. But
what's a cyanobacteria, first of all?
yeah,
That's easy.
Well, a cyanobacterium would be a bacterium that's capable of photosynthetic activity. And
Okay.
actually, some of the very earliest life forms on the planet are cyanobacteria. So these are photosynthetic bacteria that generate oxygen.
And they're not simple organisms.
No, very complex. They're
Yes.
bacterial organisms. Now, some cyanobacteria actually also fix nitrogen, or they will take nitrogen from the atmosphere and convert it into ammonia. And that's a huge thing in ecosystems because you need a source of nitrogen that can then be used to build amino acids and nucleobases. And you know, to convert nitrogen into ammonia is a very complex chemical process because there's two nitrogen atoms that are triply bonded. It's an inert molecule and it's a very strong chemical bond. And so there's an enzyme called nitrogenase that does that, and we'll get to that in a second. But this is a picture of the organelle that was originally thought to be an endosymbiont.
Can you tell us what an organelle is?
Yeah, well, an organelle is a membrane-bound structure inside a cell.
It's inside, it's not the cell itself.
No, it's inside the cell.
Yeah.
And an organelle means mini organ. So it has a dedicated and devoted biochemical function that contributes to the cell. And so people thought, well, this organelle was actually at one point an endosymbiont, and that an endosymbiont would be a microorganism that becomes engulfed by a eukaryotic cell that establishes a permanent relationship, a permanent symbiotic relationship where that bacterium now becomes a permanent residence inside the cell. So when the cell divides, the bacteria divides, you know, and is retained with it.
So it's a big microorganism incorporating a small microorganism.
Right, right. So the thought was that in this particular case, that this is an organism that's nicknamed UCYN-A, and the actual scientific name of the endosymbiont is, if I can even pronounce it here, Candidatus atelocyanobacteria thalassa. So that's why we're calling it.
That's why they came up with the acronym.
Yeah, UCYN-A.
Sounds like what happens in astrophysics.
Yeah, right. And so it's a permanent resident in a particular, algae, a eukaryotic organism that not only is involved in carbon fixation, but nitrogen fixation. And this is a organism that's distributed throughout the world's oceans. It's the primary producer of fixed nitrogen in ecosystems, in aquatic ecosystems. So it's a very important organism. But the fact that it's now that what was thought to be an endosymbiont is actually understood as an organelle is considered to be, again, an observation of the endosymbiont hypothesis. And so here's a slide showing one version. There's all kinds of versions of this hypothesis, but the idea in a nutshell is that the origin of eukaryotic cells, complex cells with a nucleus, with organelles, internal membrane systems, began when a larger bacterium of some sort or an archaeal
Not
cell.
a eukaryotic, but
No.
Yeah, a parakaryotic.
You know, and in this particular diagram, began to form these internal membrane networks where near the center there, the networks kind of coalesced to form a nucleus, and then that cell engulfed an alpha proteobacterium that devolved to give rise to mitochondria, where it was first formed this endosymbiotic relationship, and the endosymbiont became so reduced in terms of its structure and function that it literally was an organelle now, no longer a genuine organism.
There's some irony in what you just said, Fuz. It's like there's de-evolution happening. So you got this complex microorganism basically decaying and devolving and becoming an organelle.
Right. And so presumably this event from an evolutionary perspective would have happened about 2 billion years ago, because every eukaryotic cell has mitochondria in it. So it would have happened very close to the time in evolutionary terms when eukaryotic cells emerged. And then about 1.5 billion years ago, presumably there was another endosymbiotic event where another bacterium, a cyanobacterium, was encapsulated, and this later became a chloroplast, which are the organelles in plants that engage in photosynthesis, that are responsible for fixing carbon. And so that's that evolutionary model. And what these scientists are claiming in this paper is that we really are seeing an example of that happening in real time, or in approximately real time.
So they're actually claiming that they have directly observed one of these events.
Well, no, they haven't.
Okay.
What they are claiming is that what we thought to be a bona fide endosymbiont, a cyanobacterium, actually turns out to be an organelle, and but it's an organelle in development. So we're seeing the process happening before our eyes. That's kind of the nomenclature. But what's interesting is in this particular, with regard to this organelle called that's now being called a nitroplast. What they've shown is that, unlike an endosymbiont, the reproduction of the cell as it goes through the cell cycle seems to be highly regulated, highly coordinated, so that the nitroplast is dividing in conjunction with the cell, as are the mitochondria and the chloroplasts in this particular organism. Now, nobody has ever been able to really study this symbiotic relationship, because nobody knew how to grow the algae in the lab. And so this research team figured out how to grow the algae in the lab, which now meant you had enough of the algae under controlled conditions that you could actually study what was going on. So they were able to image the nitroplast during the cell cycle, during, again, replication, to show this is actually a structure that's fully integrated into the cell. Now, they also noted that the genome is dramatically reduced in the nitroplast, which is what you would expect to happen in an endosymbiotic relationship. The bacteria that's been engulfed can rely on the host metabolic processes, so it just jettisons genes. It doesn't need genes for certain metabolic processes that become redundant, but yet still retains some metabolic activity that benefits the host. So there's that mutual beneficial relationship. So that genome reduction isn't surprising, but what they did note is that there's about 1,800 proteins that are produced or that are found in the nitroplast, but about 1,200 of them are actually encoded in the nuclear genome of the algae, transcribed in the nucleus, translated in the cytoplasm, and then imported into the nitroplast. That's characteristic of what you'd expect for an organelle, not for an endosymbiont. So on that basis, they argue this is really not an endosymbiont, but it's actually, again, a bona fide organelle. So that's in effect their argument. So, but it does look like, hey, wow, here's evidence that supports.
Hey, a question: did they actually observe the organelle discharging or getting rid of genes?
No, this is all inferred.
It's all inferred.
Yeah.
No.
Yeah. Well, I mean, the evidence for the endosymbiont hypothesis, and I'm most familiar with the work that's done to account for the origin of mitochondria. All the evidence for it is inferential. It's not direct evidence. So you have, mitochondria are about the same size as a bacterium, right? They have a double membrane structure like the alpha proteobacteria. They have a small genome, a small circular piece of DNA that's believed to be the remnants of its original genome. And some of the machinery in the mitochondria resemble the machinery of alpha proteobacteria. So on that basis, they argue circumstantially, again, this was an endosymbiont that was captured by the host cell. It underwent such severe genome reduction that it eventually became integrated into the cell, became part of its architecture, and transferred some of its genes to the nuclear genome. And then those genes are transcribed in the cytoplasm, or transcribed in the nucleus, translated in the cytoplasm, imported into the bacteria.
As a physicist, what surprises me is this hypothesis is 100% inference.
It's 100% inference. And with regard to the nitroplast, that's the case too.
Yes.
It really isn't that we are observing an organelle emerging,
right?
But rather the inference is that this had the same kind of origin.
We see appearances, but no actual direct evidence.
There's no direct evidence for it. So to me, what's happened is the biological community understandably made a mistake thinking an organelle was an endosymbiont, and then we now know that it's actually an organelle. And again, it's an organelle that has some resemblance to a cyanobacteria that has nitrogen-fixing capabilities, but it's not an endosymbiont. So it's all an inference. Now, this is a diagram showing what one idea for what they think happened. And so it's essentially replicating the endosymbiont hypothesis, where you had on the top this proto-eukaryotic cell that engulfs a bacteria that becomes a mitochondria. Later, we see the chloroplast emerging, where a cyanobacteria gets engulfed by that cell. And now we have this UCYN endosymbiont that was engulfed that then becomes this organelle called a nitroplast. So that's how they're envisioning this happening.
So this was published in any physics journal. They would refer to it as a just so story.
Yeah, well, it is a just so story.
Okay. Good.
Right, yeah, so it very much is. Now, to me, the problem I have with this is, number one, it is an inference. It's not an unreasonable inference, but it is an inference. There's not direct evidence for it. But superficially, when you look at the evidence in favor of it, the homologies, the similarities, okay, yeah, superficially there seems to be a case for endosymbiogenesis. But when you start looking at what has to happen, at a biochemical level for this transition to take place, it pushes the bounds of credulity. So this is, we don't know how protein transport works in the nitroplast. It's so newly discovered or recognized as an organelle, and we finally got a model system to study. That'll come, those details will come. But we do know that there's going to be similarities between the protein transport mechanism to take proteins into the nitroplast that are going to be similar to mitochondria. And I'll explain why in a minute. But for mitochondria, protein transport is extremely complex, and it's a very complex process that displays a high level of integrated complexity. So what you see here is that you have genes in the nuclear genome that code proteins that go to the mitochondria. And those genes have a region on them that's appended at the N-terminus of the protein. This is called the signal peptide. It's like the address on a letter. It tells you where the letter is going to go. So this tells you that protein is going to the mitochondria, but not only that, it tells you where it's going to go in the mitochondria. So you've got these proteins that are made in the cytoplasm with the signal sequence. That signal sequence will recognize what's called the TOM complex, the outer membrane transporter. That's what it means. It's a very massive protein complex that will recognize that protein and then will transport it across the outer membrane into the inner membrane space. There, these proteins called chaperones bind to it to keep it from folding. It keeps it in an unfolded state. Then the cell's machinery looks at the signal sequence and says, this needs to actually go into the outer membrane. So it'll transport it to a complex called the SAM that'll cleave the signal sequence, and then it'll fold into the protein complex that it's supposed to be in the outer membrane. Or if it's going to reside in the inner membrane space, that signal sequence will tell it that there'll be cleavage of the signal sequence, and it'll fold and reside in the inner membrane space. Now, if it's going to go into the matrix, it has to cross another membrane, so there's another protein transporter called TIM. This is the inner membrane transporter, massive protein complex that will transport it into the matrix, and there it'll be the signal peptide will be cleaved and it folds into the appropriate protein. If it's going to go into the inner membrane, there's another complex called TIM22 that takes care of that process. So you've got 4 transporters, or 3 transporters, and technically you have 4 membrane complexes. 2 are transporters and 2 are processors for the outer and the inner membrane. But this is a highly integrated, elaborate process that all has to be in place all at once. So the evolutionary scenario is that the endosymbiont that became the mitochondria jettisoned genes, and those genes that were necessary for the mitochondria function happened to get incorporated into the nuclear genome. And not only did they get incorporated, but as they were incorporated, there was an additional piece of DNA attached to it that would encode a protein fragment, a signal peptide, that would tell the machinery that it goes to the mitochondria and where it goes in the mitochondria. And while that's happening, you have to have the simultaneous evolution of all these protein complexes embedded in the membrane that not only can transport and process proteins, but also recognize those signal sequences appropriately. You've got to have chaperones, you've got to have peptidases. This all has to come together at once. It can't come together in a piecemeal manner. Otherwise, the endosymbiont becoming the mitochondria is useless to the cell.
Let me review this. Okay, you got these proteins. They have these little signal proteins on them that basically say, I need to go to this location.
Yes.
Then you got a protein that actually gets it to the right location. Then when it gets to the right location, it needs to be cleaved so it can be appropriately attached to that protein. And also it has to be precise timing.
Yeah.
So it's got to be timed just right.
Yeah.
And this happens with 4 different kinds of proteins.
Yes.
And all of them have to be timed just right.
Yeah.
So yeah, it's complicated.
Yeah, so it really pushes the bounds of credulity. And when you look at the literature on the origin of protein production for mitochondria, there's not good models for how this originates. It's a lot of hand waving, but there's not a good detailed evolutionary explanation.
Has anybody ever dealt with the supervisor role? There's something inside the cell that's got to be supervising all this.
Well, this is where something we talked about earlier comes into play about biochemical finite state machines, where the instructions are actually embedded in the molecules themselves, in the design of the molecules.
So that's the supervision.
Yeah.
Yeah.
But there's got to be something or someone that is designing those
To make sure.
those algorithms that are instantiated in the protein machinery to oversee all of these operations. Now, we don't know how, again, proteins are imported into the nitroplast, but we do know that it does involve a signal peptide. They call it the transit peptide. It's attached to the C terminus, not the N terminus, but it probably is, again, a signal directing those proteins to the nitroplast. But there's got to be protein transport machinery. There's got to be peptidases. So it's a similar kind of problem. For this organelle to emerge from an endosymbiont is going to involve a whole ensemble of biochemical systems that come together all at once and are operating in an integrated, highly coordinated, fine-tuned manner all at once. It's not either or, and that, to me, suggests this is design, that this is the work of a mind. And again, we would say that the similarity between mitochondria and alpha proteobacteria, or chloroplasts and cyanobacteria, or the nitroplast and what are called diazeotropes, which are organisms that fix nitrogen, is reflecting shared design, not shared evolutionary history. Now, one other thing that I think also adds to the case for design with the nitroplast is that this is a organelle that allows this cyanobacteria to fix nitrogen during the day. Now, this is really important because in cyanobacteria that fix nitrogen, they can't both engage in photosynthesis and nitrogen fixation at the same time. Photosynthesis and carbon fixation happens in the day, nitrogen fixation happens at night. The reason why is the nitrogenase enzyme that converts nitrogen into ammonia is poisoned by oxygen. There's an iron-sulfur complex that does all the work, and it binds oxygen, and the oxygen irreversibly damages that metal center. So what do these diazeotrophs do? Well, there is a clock, literally a biochemical clock called the Chi complex that will shut down gene expression of nitrogenase during the day. And what then happens is the photosynthesis, the light reactions, and particularly photosystem 2, which generates oxygen, can go to town. But then at night, when photosystem 2 is shut down and no more oxygen is being produced, the Chi clock will turn on nitrogenous gene expression. Nitrogenous is made, nitrogen is fixed, and then it's shut off during the day. So it's an alternating cycle, which is actually really pretty cool. But with this nitroplast, what they discovered is that this can actually fix nitrogen during the day because there is no photosystem 2 in the nitroplast, right? There's no photosynthetic machinery there. And in addition to that, some of the proteins that are imported are actually oxygen proteins that protect from oxidative damage. So that means nitrogen fixation can happen at the same time as carbon fixation happens, which is an important advantage in ecosystems because it allows for a greater production of nitrogen fixation.
It explains the real purpose of the organelle, because this is a place where it can happen independent of what's going on in the rest of the cell.
Right. So, where evolutionary biologists see this emergence of this organelle in evolutionary terms, it's quite reasonable to look at this from a design perspective and see the nitroplast is a very clever design that allows for, again, daytime nitrogen fixation, and that the case for the endosymbiotic hypothesis is superficially compelling, but when you start to press on the details, and there's a whole bunch of other biochemical issues for mitochondria that I could bring up that I'm not going to.
Well, I was thinking of the analogy of a very complicated factory trying to assemble a complex system, like a Boeing aircraft or a computer. This actually looks more complicated than that.
Right.
And no one would ever think that these factories are just the product of some random assembly.
Yeah.
Question I got, though, Fuz, is that what seems to be the basis for this hypothesis is the fact that they see similarities between these organelles and these microorganisms.
Right.
I think a good research thing is there's got to be some reason for that similarity. What is that reason?
Yeah.
Maybe nobody knows at this point, but it's like surely somebody must look at this and say, from a design perspective, there has to be a reason for the similarity. Let's pursue that.
Right.
And it'll probably uncover even more evidence for design.
Yeah. And I've actually written a series of blog articles over the years about the rationale for why mitochondria and the rationale for the features of mitochondria, that it actually makes sense why it would be designed that way to look similar to. So I've done a little bit of that work with mitochondria. The advantage there is we know so much about mitochondria that you have ample data to draw from.
So the case has been made for the mitochondria.
Yeah, I think so.
But we need to make a case here.
Right.
But the fact that you could make a case for the mitochondria strongly suggests a case can also be made here.
Right.
So, Fuz, this is really exciting.
Yeah, thanks, Hugh.
And I sure hope this gets into writing somewhere. I mean, this is a phenomenal piece of research you've done here.
Well,
so.
it's going to be fun because we now have the ability to learn more and more about the nitroplast. So it's going to be a lot of fun to see what people uncover over time now about its biochemistry. And I predict, as you're pointing out, that we're going to see, more and more evidence for design in this system.
Well, I know you got a lot on your plate, but I'd love to see a little booklet come out from you about organelle design.
Yeah.
And I think this might actually help the scientific community realize, wait a minute, we're overlooking something here.
Yeah.
So,
Yeah.
not that you need more work to do, Fuz, but
Yeah.
maybe you can find some biochemists in our community to take on the project. I don't know.
Yeah.
But I'd love to see something like that being produced. So,
Yeah.
okay. Got anything else on this?
No, that's it.
Okay. Well, mine's a little less complicated than what you presented, but hopefully it'll be fun too. And I think it's kind of up your alley. And this is a paper that also got published in April, so just a few weeks ago, in Nature Communications. And it's basically taking on the problem of, what happened to the magnetic field, what happened to the oxygen content in the atmosphere. And maybe we can start off with this slide that I have here. It's the only slide for the talk. But this basically shows you the oxygenation history for the early Earth, basically making the point that for the first couple of billion years of Earth's history, the level of oxygen in the atmosphere was below 1/100%.
Yeah.
So virtually non-existent. And then you have what's called the Great Oxygenation Event happening about 2.4 billion years ago, where you get a brief blip up where it goes up 2 or 3%, and over a couple of 100 million years it drops back down to less than 1%. Stays below 1%. And this is described in our books about why it jumped up, why it came back down. A lot of it has to do with the formation of continents, which you see in Genesis chapter 1, creation day 3, where God transforms the world, our Earth, from a water world to where we now have oceans and continents. That coincides with this first oxygenation event. But then the oxygen content drops back way below 1%, stays there. And roughly around 750 million years ago, 800 million years ago, it begins to slowly increase, and it goes up to about 2% at about 600 million years ago. But 575 million years ago, it has this sudden jump from 2% to 8%. And 8% for the first time allows animals to exist, big animals, not just microscopic animals, but we would call, animals that are a centimeter across or bigger. And this is the famous Avalon explosion. 575 million years ago, the Earth goes from microorganisms to these big animals. Now, they're relatively simple animals. They're basically filter feeders. They don't have digestive tracts or circulatory systems. They don't have brains and hearts like animals we're familiar with now. And for the simple reason, 8% oxygen is not enough to sustain animals that have skeletons and digestive tracts and circulatory systems. You need at least 10% oxygen. But 8% is enough for the Avalon animals. But the moment that the oxygen jumps from 2% to 8%, you have these animals immediately. There's no evolutionary history that precedes them. They just suddenly appear. And they appear the moment that the oxygen reaches the minimum level that makes it possible for them to survive. This paper refers to this, but that's not the purpose of the paper. The purpose of the paper is what caused that big oxygen jump that enabled animals that were more than microscopic to appear for the first time. And I wrote about this in this book, Designed to the Core, where I made the point that what's in the literature is that there would be these massive landslides off of the continents into the oceans, and that these huge landslides would stimulate the plate tectonics and basically accelerate what's called the deep oxygen cycle. And the deep oxygen cycle could produce this jump in oxygen. This paper basically addresses that and says, this is in the literature, but we think it's complemented, and maybe not just in a minor way, but a major way, by the drop in the strength of Earth's magnetic field. And basically, they document that it's well established that the magnetic field weakened. Now, what they do in the paper, they say, let's do an exhaustive geomagnetic study and actually try to determine when that drop happened and what was the degree of the drop. And basically, they discovered is that before the Cambrian event, if you go back like 650 million years ago, the magnetic field was about as strong as it is today, roughly half a Gauss, and had been strong for a long time. But just before the Avalon explosion, we see a huge drop in the strength of the magnetic field. And we're talking a factor of 30 times.
What would cause something like that? Is it?
That's what they're talking about. What's the cause of this?
Okay.
So, well, actually, they don't so much address the cause. I do in this book.
Okay.
Basically explain what caused the magnetic field to drop. What's new here that's not in this book is they're basically documenting the drop was quite rapid, and we're not just talking a factor of 5 times. It weakened by a factor of 30 times. So we're talking about it going from half a Gauss to less than a 50th of a Gauss. And apparently it stayed that low for several million years. And so most of the papers focused on what would be the impact of the magnetic field being that weak for that long on the atmosphere of planet Earth. And basically they're pointing out if the magnetic field was weakened to that degree for that period of time, you'd have a lot more solar and cosmic radiation impacting not just the stratosphere, but the lower parts of the atmosphere. And this would cause that solar particle radiation and cosmic radiation to dissociate water vapor in the troposphere, the stratosphere, and the ionosphere. And so this would lead, basically we're talking about breaking apart the water molecules. You got this water vapor, you're breaking it up into hydrogen and oxygen. Hydrogen cannot be retained by Earth's gravity, so it escapes to interplanetary space. The oxygen remains. And so they're arguing that this could be a major factor in causing the oxygen content in Earth's atmosphere to go from 2% to 8%. And of course, if you jump the oxygen content that much in the atmosphere, that's going to affect the oxygen content in the ocean.
Right.
Because these Avalon animals and the Cambrian animals, they're marine animals.
Right.
And so you have to oxygenate the ocean, not just the atmosphere. And if you push it up to 2%, it doesn't do hardly anything at all to the oceans. But if you jump it up to 8%, you actually do oxygenate the oceans to a sufficient degree they can actually have these animals. That's basically what this paper is talking about, is that we're thinking this could be a complementary cause. They even argue maybe it explains the entirety of the oxygen jump from 2% to 8%, but at a minimum, we know it's a big factor. It's not a minor factor, it's a big factor in causing the oxygen to jump up. And as I pointed out in Design to the Core, we really don't have the data to show that there was these massive landslides happening at that precise time to a degree that the deep oxygen cycle would produce. It's possible, you can't rule it out, and it was basically the only game in town to explain the oxygen jump when I wrote the book. What I like about this paper is saying, hey, this is another factor that we know was in play that would cause the oxygen to jump up to that degree and make possible the Avalon explosion. Now, what I found fascinating in the paper, maybe you'd like to comment on this, is that they made no mention of the fact that the moment the oxygen hits that point, you get the animals. It's not like you're waiting 50 million years for the oxygen to basically allow these animals to microevolutionary adapt to the new oxygen. These creatures show up the moment the oxygen hits that 8% level, nor do they really deal with what happens later. Not much later. 538 million years ago, we got the Cambrian explosion. And that's when the oxygen goes from 8% to 10%, a sudden jump like this graph observes, but the moment it gets there, you got the Cambrian animals.
So what triggered that second jump in oxygen?
Well, the paper says perhaps this low magnetic field extended all the way to the Cambrian explosion. But that would basically anticipate a gradual rise in oxygen.
Right.
And I basically, having read the paper, say I think it's probably more complicated. Mm-hmm. They've identified a second factor that could contribute to the jump in oxygen. I still think the factor that's already in the published literature, the deep oxygen cycle being stimulated,
right,
is not going to be an insignificant factor either. And they hint at the end of the paper, maybe there's other factors we've overlooked.
Right. Now, what role would cyanobacteria play in producing oxygen in the atmosphere?
Well, as you've pointed out, we've had cyanobacteria for 3.8 billion years,
right,
and they pump a lot of oxygen in the atmosphere, and therefore we should expect a very rapid increase in oxygen. But notice in this graph, you got abundant cyanobacteria for a couple of billion years,
right,
and nothing happens to the oxygen in the atmosphere. The reason why is that you have all these metals
right,
in the crust of the earth that are starved for oxygen. The moment oxygen comes, they absorb the oxygen.
right.
It's like what happens when you take an iron tool and leave it in the backyard for a few days. It rusts.
Yeah.
What's happening is that pure iron is absorbing oxygen from the atmosphere. And the problem with the early Earth is you have all these metals in the crust of the Earth that are just soaking up the oxygen as fast as the cyanobacteria produce it. And it's not just the crust. Mm-hmm. You actually have oxygen-starved minerals in the mantle, and you got plate tectonics. So if it was only the crust, you wouldn't expect 2 billion years of nothing much happening in the atmosphere. But the fact is you got plate tectonics. We now know that plate tectonics was initiated 3.8 billion years ago. They used to think it was like 3 billion years ago, 2.5 billion. We now know it's very early. Mm-hmm. And so that's another explanation for why the oxygen stayed that low, is you got the crust being driven into the mantle.
Right.
And they're being exposed to all these oxygen-starved minerals in the mantle, which means that when it gets recycled back up at the crust, you got new crust material that's deprived of oxygen. Mm-hmm. And so thanks to the fact that we have this strong, aggressive plate tectonic activity, it takes a long time for the cyanobacteria to actually have an impact on the oxygen content in the atmosphere. And what we now realize is it takes the deep oxygen cycle to make that happen. And so when you get these huge tectonic events where you got massive landslides happening all over the world and driving this material into the ocean, into the subduction zones where the stuff gets driven down into the mantle, and there you get chemical reactions happening that produce a very sudden burst in oxygen. You get the volcanoes that push that back up into the atmosphere. That actually explains the first great oxygenation event. It's a deep oxygen cycle that really explains that blip, but what caused it was this incredible tectonic activity that was happening at that time, and therefore they also think that the later oxygenation events are tied in to the deep oxygen cycle. But none of that would matter if you didn't have a huge abundance of cyanobacteria on the face of the earth. You need that oxygen supply coming from there, but you also need to find some way. I mean, basically, the reason we got 20% oxygen in the atmosphere today, we've saturated the crust with oxygen. We've saturated the mantle, so it's able to go up to that level. Right. But in terms of what this paper is saying, I suspect things are a little more complicated, because what we do see is these jumps in oxygen are sudden, and so it's probably a combination of phenomena that explains those sudden jumps. And hey, we're going to have more Star Cells and God episodes where people are actually exploring these things and giving us more definitive answers. What I did describe in Design to the Core, the chapter on the core of the Earth, where we know that Earth began with a hot liquid iron core, where the entire core was liquid. About 600 million years ago, plus or minus a few tens of millions of years, the liquid core of the Earth transitioned into a solid core and an outer liquid core. And basically what's happening is the interior of the Earth was cooling down. It cooled down enough where a solid core was able to form. And it began very tiny, and our solid core grows by about 1 millimeter in diameter per year. So it's getting bigger and bigger. And if you wait long enough, there will be no liquid iron core. It'll be completely solid. That will shut down plate tectonics. It'll shut down our magnetic field. But hey, we're talking billions of years in the future before that happens. What I did put in the book, however, is that the transition from a purely liquid iron core to where you got a tiny inner solid core and an outer liquid core, that dramatically weakened Earth's magnetic field. So that explains why the field dropped by a factor of 30 times. Now, in writing the book, we didn't know the degree of the drop. We do knew that it went from about half a Gauss down to less than a 20th of a Gauss. But in terms of the timing and how fast and how global that was, this paper basically says, hey, it's it really did go down by that much. Not just by a factor of 5, but a factor of 30 times. So they basically published the paper making the point, the weakening is dramatic and it's real. And so, but they never addressed the cause. The cause is basically that transition. But what's interesting is the timing of that transition and the timing of the Cambrian and Avalon explosions.
Right.
So,
well, and you can't have the Avalon or the Cambrian explosion without that dramatic increase in oxygen.
right.
But technically, from an evolutionary perspective, just because you see uptick in the oxygen, even if it is dramatic, that isn't going to necessarily stimulate evolutionary changes, right? Those changes have to occur at a genetic level, and they're going to have to involve a transition from single-celled or colonial aggregates into multicellular organisms that have systems. Even in the Avalon
As you've
organisms,
written,
they're
Fuz,
crude systems.
is that the transition from animals being able to survive well on, say, 0.1% oxygen in the atmosphere, being able to function efficiently at 8%, that's not an easy biochemical transition.
No.
You got to totally change all the interior biochemistry for that to happen. And what this is documenting, it happens from a geological perspective instantly.
Yeah.
So, how do you make that happen that fast? And these changes are not minor; they're dramatic changes.
Yeah. Well, I mean, that when you're looking at these kind of discontinuities, it really looks like a creation event.
Right.
And the fact that things are coinciding, because the environment can't trigger evolutionary changes. They have to eventually happen at a genetic level, and so there's nothing that's compelling that, in evolutionary theory just because the environment changes.
Yeah, and the genetics isn't going to change that rapidly unless someone is tinkering with it.
Right.
So.
Well, because when you start going to multicellular organisms, you have to now have a completely different set of genetic regimes that are going to be responsible for cell diversification and for the formation of tissues and organs, and then integrated organ systems. And that involves, a developmental process going from a single-celled embryonic state to an adult form with millions and billions, maybe trillions of cells.
And some of these Avalon animals are 1 meter across, so they're not small animals.
Right.
So, and as you pointed out, oxygen typically is toxic for biochemical reactions inside the cell. You have to actually build in not only the mechanisms that can use this increased amount of oxygen, but the protective mechanisms to deal with the toxicity of oxygen.
Yeah.
So these are all very complicated steps. And so again, I think this would make an amazing booklet.
Yeah.
Actually bring this stuff in. So I'm always thinking of more work for you to do, Fuz.
Yeah.
So more work for me to do too. I mean, I'd like to.
Yeah.
It's like I think, I just came out with this book, less than 2 years ago. I need to rewrite this. So there's so much more new stuff. But that's what's exciting here about Reasons to Believe. The more we learn about science,
Yeah.
and the fact that it's happening literally on a daily basis.
Yeah.
These discoveries coming out.
Yeah.
So. Well, thank you for joining us today on Stars, Cells, and God. You can tell we're having a lot of fun about these new scientific discoveries that really strengthen our Christian faith. Please join the discussion in the comments below. Remember to like this video and to subscribe for more content. New episodes of Stars, Cells, and God release each Wednesday and are available here on our YouTube channel and on your favorite podcast app. Be sure to share this video with a friend, and remember, the more we learn about science, the more reasons we have to believe in the Bible as the inspired, inerrant Word God and Jesus Christ as Creator, Lord, and Savior. Thank you.